All right, BIMM 100 students, the final exam is just around the corner, so you're in exactly the right place. We're going to do a high-speed review of Lecture 16, cutting right to the chase and focusing on what you absolutely need to know to nail these questions.
So, here's our game plan. First, we'll talk about why this lecture can be a little tricky. Then, we'll dive deep into microarrays, and I mean really understand them. After that, we'll tackle viral genomes, lay out the universal viral playbook, see how viruses can actually be useful, and then wrap it all up with a solid strategy for the exam.
Okay, let's just get this out of the way. Lecture 16 is a lot, right? You've got these super complex lab techniques, and then, boom, the entire field of virology dropped on you. But here's the good news. You don't need to memorize every single detail. We're just going to distill it down to the core concepts that are most likely to show up on the final.
Okay, let's kick things off with our first big topic, microarrays. These are a favorite for exam questions, so we really want to be ready for whatever Professor Pasquinelli throws at us. Let's break down how these experiments work so you can go from raw data to the right answer every time.
So how does it all start? Well, you take RNA from two different groups of cells. A classic example is cells grown with serum versus cells grown without it. Then, and this is the key part, you convert that RNA into cDNA and you tag it with a fluorescent label. That glowing tag is how we're going to see what's going on with thousands of genes all at once.
And here's where it gets really cool, and honestly, pretty simple. It all comes down to the colors. If you see green, that gene is way more active in the without serum cells. See red? It's more active with the serum. Yellow means, it's pretty much the same in both. And if a spot is black, that gene's not even turned on. If you can remember this color code, you are golden.
Okay, so this is where theory meets practice. This is an actual clicker question from class, which is basically a giant flare telling you, hey, this is how I like to ask questions. It's not just about memorizing the colors. It's about using that information to make a sound biological conclusion.
So, let's really tear this apart. Look at option A. The genes are all yellow. That means their expression doesn't change much. A totally reasonable guess from that is that they might be controlled by the same stuff, the same transcriptional regulators. Makes sense. But now look at C. This is the kind of tricky question you need to be ready for. It says that for gene 1, which is red, the low signal in the other condition could be because the RNA is being degraded. And that is 100% correct. You have to remember, microarrays measure the steady-state level of RNA. That's a balance between how much is made, transcription, and how much is destroyed, degradation. Don't fall into the trap of only thinking about transcription.
All right, let's switch gears to viruses. When you first look at them, it's a mess, right? There's DNA viruses, RNA viruses, single-stranded, double-stranded, positive sense, negative sense. It's a lot. But don't worry, we can boil this all down to a really simple framework that makes it all click.
Just look at this table. You've got all these different starting points, all these different strategies. But do you see the pattern? They all have one destination in mind. Every single one of them has a plan to get to a plus-strand RNA. Why? Because that's the only thing a ribosome can read to make proteins.
And this right here, this quote from lecture is everything. Seriously, if you walk into that final exam and you only remember two things about viruses, make it these two. One, they have to make a plus-strand RNA so the cell can make viral proteins. Two, they have to make more copies of their genome. Every weird, complicated viral life cycle you see is just a different answer to these two fundamental problems.
So, building on that idea, even though their genomes are all different, viruses pretty much followed the same game plan to take over a host cell. There's a universal playbook that they all use to win.
Think of it like a 5-step heist. Step 1, get in the building. Step 2, hijack the factory to make your parts. Step 3, photocopy the blueprints. Step 4, put all the new copies of yourself together. And Step 5, get out and hit the next target. From the simplest virus to the most complex, they all follow this basic plan.
Now, this is where things get really interesting, and it's a perfect place for those application-style questions on the exam. We're going to look at how scientists have basically flipped the script and turned these tiny invaders into some of our most powerful tools.
I mean, how wild is this? Scientists were studying adenovirus, and when they tried to match its messenger RNA back to its DNA, it didn't line up perfectly. There were these big, loopy bits of DNA that had been cut out of the final message. That was one of the first major clues that led to the discovery of introns and splicing. A virus literally taught us about one of the most fundamental aspects of our own biology.
And here's that idea in action today. It's brilliant.
We take a virus that's really good at getting into cells, we gut it, we take out all the parts that make you sick, and we replace them with a working copy of a human gene. For something like spinal muscular atrophy, we can pack the SMN1 gene in there and use the virus like a delivery truck to get that life-saving gene to the right cells.
And it doesn't stop there. We can also use their incredible specificity. You have these viruses called bacteriophages that are like little assassins that only infect very, very specific types of bacteria. So we can put them in a spray like Listerex to kill off dangerous listeria on our food without harming us or any of the good bacteria. It's like a biological smart bomb.
Okay, let's bring it all home. We have covered a ton of ground, so let's just lock in the absolute most important takeaways for the final exam.
So here's what you need to have down cold. First, microarray results are about the steady-state level of RNA. That's production minus degradation. Second, all viruses, no matter how weird, have to solve the same two problems: make protein, copy genome. And third, be able to talk about how we've cleverly turned viruses from foes into amazing tools for things like gene therapy. If you can connect these big ideas, you'll be in great shape.
So, as you finish up your studying, here's a final thought to chew on. When you think about that viral playbook, which step do you think holds the most promise for the future of medicine? Their amazing ability to target specific cells? Their efficiency? The answer to that could very well define your careers. All right, that's Lecture 16 in a nutshell. Good luck on the final. You've got this.